Hello. In today's video, we shall learn in detail about enzymes. By the end of this video, you'll have a clear understanding about the properties of enzymes and activation energy, mechanism of enzyme action, factors affecting enzyme activity, types of inhibition, and classification of enzymes along with holoenzyme, apoenzyme, and cofactors. Let us begin with the introduction to enzymes. Enzymes are the complex protein molecules that act as biological catalysts by enhancing chemical reactions in living organisms without undergoing any change. The process of accelerating a chemical reaction is called catalysis, and the substance that speeds up the reaction is known as a catalyst. Enzymes are called biological catalyst or biocatalyst. Enzymes play an important role in cellular metabolism. They are highly specific in the choice of substrates. Substrates are molecules on which the enzyme acts. The study of the properties, structure, functions of enzymes, and enzyme-catalyzed reactions is known as enzymology. Let us understand the term activation energy. The amount of energy required to initiate any chemical reaction at a given temperature to reach the transition state is called activation energy. The activated condition of the substrate is called the transition state. For a reaction to speed up, there should be either an increase in temperature of the mixture so that the kinetic energy of the molecules increases, causing collision and reaction, or addition of enzymes which would lower the activation energy of the reaction. Enzymes bind to reactant molecules and help in the breaking of old bonds and formation of new bonds, resulting in product formation. Enzymes act by stabilizing the transition state. The enzyme lowers the activation energy of the reaction and speeds up the reaction. The reactant molecules absorb thermal energy from their surrounding, which helps to speed up the motion of molecules, increasing the force of their collisions, resulting in breaking of bonds. An enzyme releases the activation energy needed for a reaction to occur. It provides the reactants with better orientation so that the frequency of collision increases. This helps in increasing the rate of reaction. With the formation of new bonds, products are formed. Enzymes are released and can again combine with another molecule of the substrate. By the way, if you're getting some value out of this video, please like and share this video so that all of us can learn, unlearn, and relearn together. Also, please consider subscribing the channel and press the bell icon to get instant notification of all the upcoming videos. Now, let us learn about the properties of enzymes. Enzymes behave as colloids and have high molecular weight. They help in accelerating the rate of biochemical reaction but do not affect the nature of the final product. Enzymes remain unchanged at the end of the reaction. The enzyme-controlled reactions are reversible. They accelerate the forward or reverse reaction to attain equilibrium. Most enzymes have high turnover number. Turnover number is the number of substrate molecules acted upon by one molecule of enzyme per unit of time. Enzymes are said to be efficient when the turnover number is high. The enzymes are specific in action. A given enzyme catalyzes only one reaction or a similar type of reaction, or may act on a particular substrate only. Enzymes are made up of long chains of amino acids. The amino acid sequence gives enzymes their substrate specificity. Enzymes are heat sensitive. Enzyme activity decreases with increase in temperature. Enzymes perform well at an optimum temperature of 25 to 40 degrees Celsius. They become inactivated at very low temperature and gets denatured in high temperature. Enzymes show maximum activity at an optimum pH of 7.5. Enzymes possess active sites with which the substrate interacts to produce the desired product. Enzymes are required in small concentration for any chemical reaction. Velocity of an enzyme increases with an increase in substrate concentration. Enzymes are sensitive to inhibitors. They are destroyed by poison or drugs such as cyanide, which destroys the respiratory cytochrome enzymes. Next, let us understand the mechanism of enzyme action. Two different models have been proposed to explain the mechanism of enzyme action. They are lock and key model and induced fit model. Lock and key model was proposed by Emil Fischer to explain the enzyme-substrate interaction. Every enzyme-substrate interaction possesses specific geometric shape which are complementary to each other and therefore fit exactly into one another. A small fraction of the enzyme, known as the catalytic site, is involved in catalysis by reducing the activation energy. The other portion for binding with the substrate is known as the binding site. The catalytic site and the binding site together constitute the active site of an enzyme. The substrates have reactive sites on their surface to fit the active sites of enzymes. The active sites and the reactive sites fit like a lock and key. Enzyme represents the lock and the substrate represents the key. The enzyme molecule lowers the activation energy. The substrate and the enzyme is known as the enzyme-substrate complex. Once the product is formed, it leaves the enzyme surface as it can no longer fit into the active site. The enzyme molecule is now free to join with another substrate molecule. Next model is the induced fit model. Koshland suggested that the active site continues to change its conformation or structure until the substrate gets tightly bound to the enzyme at the active site. In the highly unstable transition state, the old chemical bonds break and the atoms rearrange themselves and new bonds are formed. Binding of the substrate molecule to the enzyme induces a conformational change in the enzyme. Protein molecules of the enzyme are flexible, thereby allowing the conformational change of the active site. The tight fit conformation and lowering of activation energy allows the reaction to proceed readily. The conformation of the active site in enzyme is influenced by many factors like coenzymes, temperature, cofactors, etc. The product formed leaves the enzyme. The enzyme now receives another set of substrate molecules. Next are the factors affecting enzyme activity. There are various factors influencing the rate of the enzyme catalyzed reaction. Enzyme concentration. There is always a steady increase in the rate of an enzyme catalyzed reaction with an increase in the number of enzyme molecules, provided there is enough concentration of substrate. Substrate concentration. There is an increase in the activity of enzyme with an increase in the concentration of substrate. When the active sites of the enzyme molecules are bound or saturated with substrate, any increase in substrate will have no effect on the rate of reaction. Product concentration. Accumulation of the product lowers the enzyme activity. Temperature. At 0 degrees Celsius, the activity of enzymes is minimum or zero. With increase in temperature, there is increase in enzyme activity up to a certain optimum. Enzymes show maximum activity between 25 to 40 degrees Celsius. After 45 degrees Celsius, enzyme activity is retarded and it stops altogether beyond 60 to 70 degrees Celsius. As the temperature rises, the kinetic energy of the molecules increases and the weak hydrogen bonds break, disturbing the tertiary structure of the enzyme. This change in structure is known as denaturation of protein. Denatured enzyme protein remains inactive and further increase in temperature destroys the protein completely. Hydrogen ion concentration. Enzymes are active only over a limited range of pH. The maximum activity of enzymes is seen in a pH range of about 6 to 7.5. Decrease in enzyme activity due to shift towards alkaline side or acid side causes denaturation of the enzyme molecule. Inhibitors. Certain substances or chemicals prevent an enzyme to function. They are known as inhibitors. Different types of inhibition are competitive inhibition, non-competitive inhibition, and allosteric inhibition. Competitive inhibition. A substance shows a close structural similarity or resemblance to the substrate, due to which both compete for the same active site of the enzyme. Such a substance is known as an inhibitor. It forms enzyme inhibitor complex, hence there is no resultant product. Example, due to structural similarity between malonic acid and succinic acid, malonic acid inhibits the activity of enzyme succinic dehydrogenase, which catalyzes the conversion of succinic acid to fumaric acid. Competitive inhibitors play a major role in controlling bacterial pathogens. The competitive inhibition of enzyme activity is a reversible reaction. In the presence of excess substrate, the inhibitor molecules are removed and the enzyme becomes active again. Non-competitive inhibition. In this case, a substance does not resemble the substrate in structure and therefore there is no competition for active site of enzyme. Instead, it binds to the enzyme at some other site, due to which the physical structure of enzyme is changed and no product is formed. Example, cyanide inhibits cytochrome oxidase, which is required for cellular respiration. Non-competitive inhibition of an enzyme is irreversible. Addition of substrate does not restore the enzyme activity. Allosteric inhibition. Certain substances attach to an enzyme at a specific site called allosteric site and change the structure of the active site. Such substances or inhibitors are called modulators. It does not allow the substrate to bind to the active site of the enzyme, thus hindering the reaction. The process is called allosteric inhibition, and the enzymes with allosteric sites are called allosteric enzymes. Example, in respiration, during glycolysis, enzyme hexokinase acts on glucose to form glucose 6-phosphate. Accumulation of the final product inhibits hexokinase to carry on with the reaction. The product acts as an inhibitor, causes conformational changes in the active sites of the enzyme, and brings down the catalytic activity. This is called feedback allosteric inhibition. Activators. Certain enzymes are in the inactive form within the living cells. They are called zymogens or proenzymes. They are activated by specific ions or other enzymes and are called activators. Example, inactive pepsinogen in the gastric juice of stomach is converted into active pepsin by hydrogen ions. Poisons and radiation. The tertiary structure of enzymes are destroyed by radiation and poisons like cyanide, due to which the enzymes become ineffective. Next, let us learn about the classification of enzymes. Based upon the type of chemical reactions they catalyze, enzymes are divided into 6 major groups. Oxidoreductases. These enzymes catalyze oxidation and reduction reactions. Example, phosphoglyceraldehyde dehydrogenase oxidizes phosphoglyceraldehyde. Transferases. Transfer of groups from one substrate to another other than hydrogen is catalyzed by transferases. Example, hexokinase transfers a phosphate group from ATP to glucose to form glucose 6-phosphate. Hydrolases. These enzymes catalyze splitting of molecules or breaking of bonds by addition of water. All the digestive enzymes are hydrolases. Example, amylase hydrolyzes starch and glycogen to maltose. Lyases. These enzymes catalyze the breakdown of chemical bonds without hydrolysis. Example, aldolase splits fructose 1,6-diphosphate into glyceraldehyde 3-phosphate and dihydroxyacetone phosphate. Isomerases. These enzymes catalyze the interconversion of isomers. Example, glucose 6-phosphate is converted into fructose 6-phosphate with the help of phosphohexose isomerase enzyme. Ligases. These enzymes catalyze the union of two compounds with the help of energy from hydrolysis. Example, during carbon dioxide fixation, pyruvate carboxylase unites pyruvate and carbon dioxide to form oxaloacetate. Based on the site of action, enzymes are of two types: intracellular enzymes and extracellular enzymes. Intracellular enzymes are produced by the cell. They remain and function inside the same cell. They are also known as endoenzymes. Example, enzymes of glycolysis and TCA cycle. Extracellular enzymes. Enzymes produced by a cell but act or function outside that cell are known as extracellular enzymes or exoenzymes. Example, salivary amylase secreted by the cells of salivary glands and pancreatic lipase secreted by the pancreas. Isoenzymes or isozymes. Enzymes that catalyze the same type of reaction but have different molecular structure are known as isoenzymes. Example, alcohol dehydrogenase has 4 isoenzymes in maize. Cofactors are the non-protein molecules. It is bound to the enzyme to make the enzyme catalytically active. The inactive protein part of the enzyme is known as apoenzyme. A combination of an apoenzyme and cofactor is called holoenzyme. An organic cofactor which can be easily detached from the apoenzyme is called coenzyme. They are often vitamins or derivatives of vitamins. Prosthetic group. The cofactor that is tightly bound and cannot be separated from the enzyme is termed a prosthetic group. Example, the heme of hemoglobin. So today we have learnt in detail about properties of enzymes, activation energy, mechanism of enzyme action, factors affecting enzyme activity, classification of enzymes along with holoenzymes, apoenzymes and cofactors. I have some practice questions for you. Please share your answers in the comments section below. You can pause the video and write down the questions if you like. Thank you so much for your time and participation. If you haven't subscribed to my channel yet, please consider subscribing the channel and press the bell icon to get instant notification of all the upcoming videos. Also, if you want to discuss any particular topic in biology, please mention that in the comments section below. I'll see you there. Goodbye. All the best.